Hello everyone, myself Dr. Harsha. Today I am going to start a new topic. That is, I am going to discuss today inborn error of metabolism. So before starting this topic, I'll share a small story with you. Just imagine you're all sleeping, you have got a dream. In your dream, you're traveling to Mars. You're taking a trip to the Mars. Elon Musk has already invented the rocket and he is taking a trip. Whoever is having enough money, they are going to Mars. So when you landed on Mars, what you saw is, you saw rivers, oceans, you saw few creatures also there. So those creatures are almost similar to us. Some difference was there. What was that difference is, they were having a long nose, but it was not in front of the face, it was on the back. So what you thought is, you want to decode, they were speaking some different language. Because you have spent so much amount, you just don't want to come back without interacting with those aliens. So as it is not a live class, you can comment in the chat box how many of you feel this IM topic as alien. So including myself, even I feel this topic as alien. So now you want to decode this alien. Okay. So how you will decode? So in order to decode this topic, you need to learn you need to already have learned, you need to refresh and recall your basics of biochemistry. Don't feel bad. You have already left the biochemistry subject in your first year. Now you are feeling okay, it has followed till your post-graduation. So don't feel sad. So we'll not learn in depth, but we will have some basic knowledge. So let's start the topic. So first we will start with glucose homeostasis. So what is going to happen whenever you have a meal or whenever you consume food? What is going to happen initially? There is a raise in the blood glucose level. So after some time, so what is going to happen? After 2 to 4 hours, the glucose level will come down. So whenever there is a raise in the blood glucose level, there is a increase in the secretion of hormone insulin. Insulin is secreted. So what is happening? Insulin is secreted. Glucagon is in the decreased, matlab it is in a small amount, but there is an increase in insulin hormone. So because there is so much of glucose you are consuming, the glucose is converted into glycogen. Glycogen is nothing but a polymer of glucose. Because there is excess, now you are storing. Then because you are consuming food, what you are doing, there is so much of glucose. What you are doing, there is a lipid synthesis, protein synthesis. So there is a storage. After 2 to 4 hours of meal, what is happening? The glucose level will come down. So when the glucose level comes down during the fasting state, the insulin hormone will decrease and counter-regulatory hormones like adrenaline, glucagon, cortisol and growth hormone, what they'll do? They'll increase. So the rescue team is what now? First rescuer is glycogen. So in the early phase, there is a glycogenolysis followed by gluconeogenesis followed by lipolysis. So this is the same thing. So there is a first initially 2 to 4 hours there is a increase in the glucose. After that overnight. So in Nelson they have not mentioned specific timing. So when till what time the glycogenolysis will predominate. So overnight fasting. So when you consume food and sleep, after 4 hours the overnight fasting is taken care by glycogenolysis. Suppose overnight fasting, just think 8 to 12 hours. Then what happens? The glycogen storage will get depleted over a period of time. Then what is going to happen? Gluconeogenesis will set in. How the gluconeogenesis occur? The source of gluconeogenesis are different. For example, you can make glucose from amino acids also. Like when the amino acid metabolism occurs, the carbon skeleton will also contribute to formation of glucose. So that will we will discuss in the further slide. So the gluconeogenesis will take over. So if there is a still prolonged fasting, the child has not consumed anything, what is going to happen? There is a lipolysis. That is, fatty acid oxidation will predominate for the energy source. Ketone bodies will act as a source of energy for prolonged fasting. So this is all the similar thing which I have depicted in a picture. So fed state, there is an increase in insulin, there is a glycogenesis, lipogenesis and protein synthesis. Early fasting phase, decrease in insulin, increase in glucagon, glycogenolysis take place and gluconeogenesis will start. Late and prolonged fasting, glycogen depletion is there. Then gluconeogenesis still trying to maintain the glucose, lipolysis and ketone body are the source of fuel for the body. So what is going to happen when we consume, whether we are consuming protein, whether we are consuming fat or whether we are consuming glucose? The end or fate of everything is what? Everything is getting converted into acetyl-CoA. So glycolysis. So glucose is there. Glycolysis is happening in the cytosol. Pyruvate. We all know that pyruvate, with the help of enzyme, pyruvate dehydrogenase enzyme, pyruvate is converted into acetyl-CoA. Fatty acids, fatty acid beta oxidation will occur in the mitochondria. This also produces acetyl-CoA. Protein metabolism, amino acids. We all know that building blocks of protein are amino acids. So during the metabolism of amino acid, what is left? Ammonia is produced and the carbon skeleton is converted into acetyl-CoA. So this is the main thing, main source of energy. This acetyl-CoA is going to enter our Krebs cycle. That is what we call as TCA cycle, where this acetyl-CoA enters the mitochondria and it produces ATP. So this is the Krebs cycle. So briefly we'll learn about Krebs cycle. Acetyl-CoA is a 2-molecular compound. It combines with oxaloacetate, which is a 4-molecular compound, and it produces citrate. Citrate is converted into isocitrate. Isocitrate is converted into alpha-ketoglutarate, where there is a conversion of NAD plus to NADH plus. So there is a formation of 2.5 ATP. Then this alpha-ketoglutarate is a complex enzyme which requires all these factors. Then this is converted into succinyl-CoA. So here also there is a production of NADH. So CoA, thymine pyrophosphate and lipomide. So this is produced. This is also producing 2.5 ATP. Then succinyl-CoA. Succinyl-CoA is converted into succinate. Succinate into fumarate. So when succinyl-CoA is getting converted into succinate, it produces 1 ATP. Here there is a production of FADH2. Here there is a production of 1.5 ATP. Succinate to fumarate. Then fumarate to malate. Malate to oxaloacetate. Here 2.5 ATP. So when you count all this, so this Krebs cycle with acetyl-CoA, it is producing 10 ATP. What is ATP? ATP is nothing but a source of energy, fuel. Okay. So we all know that this acetyl-CoA is taking place, this Krebs cycle is taking place in mitochondria, which produces energy. So when we were in school, we used to learn mitochondria is the powerhouse. So fuel. So this is what? This is glycolysis cycle. So why I am discussing here glycolysis is, just now I told gluconeogenesis is going to happen. Even amino acids produce glucose, which is what we call as gluconeogenic amino acids. Glycolysis is a pathway where there is a degradation of glucose. This pathway can also be reversed to produce glucose in what? In fasting condition. Here pyruvate is there. Pyruvate can be, again it can be this cycle, this pathway can be reversed and we can produce glucose molecule whenever there is a fasting. So in general what happens, whenever there is a pyruvate production is there with the help of enzyme, pyruvate dehydrogenase enzyme whenever there is a oxygen molecule. So in case of aerobic metabolism, there is a production of acetyl CoA which enters Krebs cycle. Whenever there is a shock, suppose shock is there, a child has come with suppose a severe shock. What is going to happen in case of shock? There is no oxygen or decreased oxygen. At that time, what is going to happen? The energy is produced by anaerobic metabolism. Pyruvate is converted into lactate. That's why in case of shock, you are having what? Increase in lactate levels. So glycolysis is the only source of energy in case of erythrocytes. And this pathway occurs in most of the cells. And it also provides carbon skeleton for this glycolysis pathway, also provides carbon skeleton for non-essential amino acids. What are non-essential amino acids? Which can be produced, the amino acids which can be synthesized by our body itself. Now coming to amino acid metabolism. So amino, this is the structure of an amino acid where you have a amino group and you have a carbon skeleton. So this is a amino acid. So how during metabolism, what is going to happen? This ammonia is removed and this carbon skeleton is transported for further things. For example, this carbon skeleton can be converted to pyruvate. It can also be converted to intermediate compounds of Krebs cycle. It can be converted to acetyl CoA and acetoacetyl CoA. So this is an overview of amino acid metabolism. So this is an amino acid, this is a keto acid. What is happening? You have an amino group and a keto group. So first thing is what is happening is transamination. So what do you understand by transamination? You are removing this amino group and adding this amino group to the keto acid. So what is happening? This will form a keto group. So alpha ketoglutarate, and this will form a new amino acid, that is glutamate. So after transamination, there is something called as deamination. So again you are removing the ammonia. So what is happening? So there is a removal of ammonia group and glutamate is converted into alpha ketoglutarate. So alpha ketoglutarate. Then after deamination, what is there? After deamination, now you have got ammonia, free ammonia. What is happening? This free ammonia is very toxic whenever it circulates freely in the blood. So this ammonia combines with another molecule of glutamate. In the presence of enzyme glutamine synthetase to form glutamine. So now this glutamine is traveling in the blood and it reaches liver where detoxification occurs. So again with the help of enzyme glutaminase, ammonia is released in the liver. So in liver, what is happening? Detoxification of the ammonia, it is converted into urea. This urea with the help of kidney excreted in the urine. So this is the overall view of amino acid metabolism. Next, I have already told there are few amino acids. So I have told here there is what? Carbon skeleton left here during amino acid metabolism. Now what is happening? This carbon skeleton of few amino acids, what they will form? For example, isoleucine, methionine, valine and threonine, they are going to, after the metabolism of these amino acids, they are going to produce succinyl CoA. So again this Krebs cycle continues. Phenylalanine and tyrosine can be, after metabolism, they can be converted into fumarate. Then arginine, glutamine, histidine, they can be converted into glutamate. Again glutamate can be converted into alpha ketoglutarate. Lysine and leucine can be converted into acetoacetyl CoA, that is ketone bodies. So few isoleucine, leucine, tryptophan, they can be converted directly into acetyl CoA and these amino acids can be converted into pyruvate. So there are some amino acids after metabolism, they can be converted into gluconeogenic and ketogenic thing. So from this glucose also can be produced and ketone bodies can also be produced. There are some amino acids after metabolism, they are going to produce only ketone bodies, that is leucine and lysine. So see, this we all know that this TCA cycle is a very important cycle. It is like the cycle is continued. All the products which are required for the functioning of TCA cycle are supplied by the amino acids after metabolism. It is like suppose you are cooking in a kitchen, it is a supply chain. You need cereals also, you need pulses also, you need vegetables also. So when you are cooking, all the supply. So this is an important powerhouse. Cooking food, again energy. So you compare with a kitchen. So all those things are supplied by what? All these things. So that the TCA cycle can smoothly function and we get energy. Next is amino acid metabolism. So this is already I have discussed amino. So this is the same thing where you can know what all things are converted into what. Next is, this is the last one, beta oxidation of fatty acids. So we all know that fatty acids, beta oxidation of fatty acid, again it is going to produce what? Acetyl CoA. So this is the metabolism of fatty acid. So usually there are so many types. Beta oxidation. In beta oxidation there are even chain fatty acid, there are odd chain fatty acid. So the metabolism, the yielded product is different. For even chain fatty acid, there is a production of acetyl CoA. So acetyl CoA, fatty acid has to be activated in the cytosol, that is fatty acyl CoA. The activated fatty acid, they enter what? Mitochondria. They cannot enter mitochondria, they need some help, that is carnitine palmitoyl transferase 1, which is present in the outer surface of the mitochondrial membrane. So there is something called as translocase enzyme. So again from here it is transferred to the activated fatty acid are transported here. From here they are transported into the inner mitochondrial membrane with the help of carnitine palmitoyl transferase 2. Now it has entered mitochondria where beta oxidation of fatty acid is going to take place with a series of process. First is dehydrogenation, hydration, then dehydrogenation, then thiolysis. See what you are seeing is there is a production of acetyl CoA. Then this acetyl CoA is going to enter the Krebs cycle for energy production. So what is the significance? What is going to happen? See, just now why we are reading all these things is, suppose there is a problem. For example, I have told during fasting state, what is required? Glycolysis is the first rescue mechanism. For example, the baby is 4-month-old baby. Whenever it is fasting for about suppose 8 hours or 9 hours, whenever it is sleeping, it is getting hypoglycemia in the early morning. So at that time you can suspect there could be a defect in what? Defect in glycolysis. That's why there is no production of what? Glucose. For example, if there is a prolonged fasting, what is the fuel of rescue? That is fatty acid oxidation should occur. If on prolonged fasting, if the child is having hypoglycemia, there could be a defect in fatty acid metabolism. So there is some link. For example, if there is a defect in the amino acid metabolism, so many products are formed. Toxic products are formed which could be harmful to the brain. They are toxic to the brain. So that's why you have to learn these things in bit detail. There are so many types of oxidation. For example, even chain fatty acid, it is taking place in mitochondria, it is yielding acetyl CoA, then energy production. Odd chain fatty acid, it is taking place in mitochondria. Odd chain means what? Even chain is, for example, 4, 2, 8, 6, 12. So even number. Odd chain fatty acid, what they are yielding? They are yielding propionyl CoA. This propionyl CoA is converted again into succinyl CoA, which enters again already you know succinyl CoA is an intermediate product of TCA cycle. So energy production is there. Suppose there is a problem in even chain fatty acid. What is going to happen? I have already discussed the cycle. So there are so many types. So what is going to happen? Medium chain acyl dehydrogenase enzyme deficiency, carnitine deficiency, carnitine transporter deficiency, and Jamaican vomiting syndrome. Odd chain fatty acid, I told there is a production of propionyl CoA to succinyl CoA. So if there is a deficiency of propionyl CoA carboxylase, there is a accumulation of propionyl CoA. There is something called as very long chain fatty acid. What is happening in this? It is taking place in peroxisome. Because it is very long. Long chain fatty acids are nothing but those which are having carbon atoms more than 22. So the main function of peroxisome is to shorten the fatty acids so that a better production, matlab, to attain a good size, and it should go to mitochondria and produce ATP. So if there is a defect in the shortening of this very long chain fatty acid, what is going to happen? There is something called as Zellweger syndrome and adrenoleukodystrophy. There is something called as alpha oxidation. The alpha oxidation, it occurs with the fatty acids with methyl group at beta carbon. Now just think, there is a fatty acid. They are having what? Methyl group. What is methyl group? It is CH3. So beta carbon, for example, at the beta group, so there is something called as alpha, there is something called as beta. So at beta carbon atom, there is a methyl group. So these kinds of fatty acids, they undergo what? They undergo oxidation in case of these things, that is taking place in what? Endoplasmic reticulum. Not only endoplasmic reticulum, some amount is going to take place in peroxisome also. So if there is a defect in this, what is going to happen? There is a disease called as Refsum's disease. So this alpha oxidation, example is what? Phytanic acid, which is produced in the milk. If the child is having Refsum's disease, you should avoid milk. Omega, this is not implicated in any disease, so we will not discuss. So actually I have started a series of IM. This was a video of basics of IM. So now in the next class I am going to start a discussion on organic acidemia. So all this link I will provide after finishing all those IM series. I am going to provide a link for this note. So thank you for the patient listening. ಎಷ್ಟು ನಿಮಿಷ ಆಗಿದೆ ನೋಡಪ್ಪ.
21
ನಡೀತೈತೆ. 20 ಇಸ್ ಆಲ್ಸೋ ಫೈನ್.
1 ನಿಮಿಷ ಹೋಗುತ್ತಾ
ಎಲ್ಲಿ ಆ ಕಡೆ ಹೋದ್ರೂ ನಡೀತಿತ್ತು. ಫಸ್ಟ್ ವಿಡಿಯೋ ಐತಿ. ಆಮೇಲೆ ಭಾಳ ಬಡ ಹೇಳಿದ್ರು. Hello everyone, myself Dr. Harsha. Today I am going to start a new topic. That is, I am going to discuss today inborn error of metabolism. So before starting this topic, I'll share a small story with you. Just imagine you are all sleeping, you have got a dream. In your dream, you are traveling to Mars. You are taking a trip to the Mars. Elon Musk has already invented the rocket and he is taking a trip. Whoever is having enough money, they are going to Mars. So when you landed on Mars, what you saw is, you saw ಇದು 5 minutes actually ಕಥೆನೇ ಅದ.
You saw few creatures also
ಅದು
there.
ನನ್ನ ಕಥೆ